Okay. All right. So just a brief introduction. I'm Megan Salinas. I'm one of the movement disorder neurologists. I've just joined Rocky Mountain Movement Disorder Center in Inglewood in the summer. And so have relocated from very hot Dallas, Texas, and have been enjoying myself immensely with the mountain views and the cool air. Um, so I will jump right in. And I think Delisa also deserves another thank you for getting all of our heart rates up and our blood running so that I can put you to sleep. No. All right. Well, I turned it off and then back on. Okay. All right. So we're going to go over a few important topics today. So we're going to discuss the research pipeline. Most of my patients will ask me, usually they wait to the end of the visit and they're like, one more question. What's coming out? What's happening next? Is there, a brilliant treatment right around the corner? So we'll talk about that process so you can educate yourselves and understand that kind of lingo. We'll talk about alpha-synuclein, which is the abnormal protein accumulation. Thank you so much. And Parkinson. We'll talk about some drug targeting strategies based on the alpha-synuclein protein, as well as briefly mention a few current clinical trials and what their targets are. Okay, we're in business. Okay, so why should someone such as yourself or a family member participate in clinical research? Well, there's many different reasons. The first one is to have access to innovative treatments. This will be much more clear in a few slides when we talk about the research pipeline, but it can take years, over a decade, to get a new drug to market with FDA approval. And so, as you know, with Parkinson's, the disease is progressive. And so if you're wanting to have access to the latest treatment options or disease-modifying therapies, potentially, having an involvement in a clinical trial can provide those for you. All right. You can also contribute to medical progress. So helping scientists, physicians understand Parkinson's disease, the physiology, what's happening in the body, biomarkers, so what blood tests, imaging tests, so we can diagnose the condition earlier and earlier, as well as determining if there's a new treatment target so that they can develop drugs or other treatments that work differently than what's available currently. When you're in a clinical trial, you, depending on the trial, can have frequent clinical visits. So with that, you are followed closely. And so if your disease progresses, or if you develop new symptoms, motor, non-motor related to Parkinson's, then you know that you have a whole team equipped to help you tackle those problems. And lastly, hope for yourself as well as future generations. So again, that gets to the exciting time that we have with Parkinson's disease and many other neurodegenerative conditions. We are starting to go into the later phases and early phase clinical trials of disease-modifying therapies. So we don't know that they work yet, but we have a lot of encouraging data. If you were to have asked me 5-10 years ago, it was less promising at that time. So this is really an exciting time to be involved with research. And of course, for future generations. So some Parkinson's disease does run in families. There's also genes that you can inherit that puts you at risk for Parkinson's, but doesn't necessarily cause it. We'll talk a bit more about that later. But you can think of your participation as informing the science so that if your future generations, your great grandkids, so on, will have treatments that maybe you weren't immediately able to benefit from. All right, so there's different types of clinical research. And so not all clinical studies are in fact interventional trials, meaning they're not giving you a drug or compound that hasn't been studied very long in humans. So you have observational studies. So again, this does not have an intervention. It's following disease progression over the years, and it may be in different population, men versus women, different ethnic backgrounds, those sorts of things. Also different genetic mutations. Of course, we have genetic studies, which is listed third here. So kind of figuring out which genes may put individuals at risk for Parkinson's. Well, how do we get that information? Hundreds and thousands of individuals participated in clinical trials, allowing geneticists to study their DNA to find these gene mutations. And I'll talk about in a minute, this has greatly informed science so that we could find targets to treat Parkinson's and hopefully slow it down. Interventional studies, so they're certainly disease-modifying therapy. You'll hear me say that over and over again, mostly because I'm very excited about it, but also symptomatic treatment. So symptomatic treatment is similar to carbidopa-levodopa. How many of you guys have heard of that? Or the brand name, which I'm not supposed to say here, but essentially that is a symptomatic treatment. It does not alter the disease course, meaning it will not slow or hasten the disease progression. So there's symptomatic drug testing trials. You can also have devices. So there's different devices for tremor, let's say, that can help offset tremor for a few hours. Deep brain stimulation. So back in the mid to late 90s, there was a lot of deep brain stimulation trials to determine if that worked. Fast forward, it did, and it's one of our very effective treatments. All right, biomarker research. So this goes really hand in hand with observational studies. So in order to cure Parkinson's or significantly slow down disease progression, we need to catch Parkinson's almost before it causes any symptoms. By the time someone has motor symptoms of Parkinson's, about 60% of their dopamine producing brain cells have already died back. That's quite a lot. So if we're going to implement a treatment to alter that trajectory, we need to do it as early as possible. And so finding blood tests, imaging tests, biopsies that can inform us of a diagnosis before we can make that clinically will allow us to test interventions at a very early time. Quality of life research. So this also goes with Delisa's exercise. So it's not just looking at symptomatic control of motor symptoms of Parkinson's or slowing down disease progression. We want to make sure that we can improve your day-to-day quality of life. There's lots of ongoing studies in music therapy, dance, boxing, those sorts of things in Parkinson's. All right. So I get asked lots of questions about this. The placebo in clinical trials. So essentially a placebo is given in lieu of the investigational drug so that researchers can tell if the investigational drug worked. What's important to know, and many may have already heard this saying, is that there's a placebo response. And so there's a psychological impact of being involved in the trial and being hopeful that the trial will work or that you will receive the investigational product in the trial. And so people actually will have their neurologic symptoms improve a little bit. This is not unique to Parkinson's. In all neurologic conditions combined, the placebo response can be as high as 30% improvement. That's a lot. So then we have to make sure that our investigational drugs are better than that, right? Okay, it also helps us monitor for side effects that are unique to the investigational drug, okay? All right, so ethical considerations. So if someone is needing a life-saving treatment, not necessarily applying to Parkinson's, but let's say you go to the emergency room and you have a heart attack and they have a clinical trial that could save someone's life that otherwise would not be saved, then that would not be an ethical situation to give that person placebo, right? So in some studies, if it's a higher risk, they may not offer placebo because the benefit of the investigational drug is high enough, okay? There's trials that have open label extensions. What does that mean? So that means that they have the certain time period of a clinical trial, and after that time period ends, they allow patients and participants to re-enroll to receive the investigational drug, okay, or treatment. So with that, even those who are receiving placebo during the actual study time will then receive the investigational product. So we always counsel our patients that are interested in wanting to learn, and they're like, well, what if I get placebo? Well, one thing that offers hope is that, well, even if you're in the placebo group that doesn't receive the investigational treatment, you have the option to sign up and re-enroll to participate in the open label extension when you know that you'll receive it. And so especially with these disease modifying therapy trials, that's very important. All right, I think I covered. All right, so the pipeline. So there's different stages. So you have preclinical testing. Even before a compound or drug makes it to preclinical testing, years, handfuls of years are being spent in a laboratory screening medications or compounds to find something that may work. Then they take that compound in the lab and they test it. So this is where they are testing it on, human cells that they've banked or animal model research to see if it is safe and if it works as they intended without side effects, okay? Once they find that they have good effect in their models and acceptable level of side effects, then they can apply to the FDA for an investigational drug status. This means that the FDA reviews all of the animal model or non-human data to make sure that it makes sense, that the benefit to risk ratio is in a person's favor, okay? We don't want the treatment to be worse than the problem, right? So once the FDA clears a drug, it goes into phase 1 testing. So phase 1 trials are small. Often these are comprised of healthy individuals, though sometimes they can be with just Parkinson patients or other conditions. So the main goal of these small phase 1 trials is to assess safety and to find at what dose the potential benefit may happen or what blood levels, brain levels that they're looking to achieve for the maximal effect. These are small trials, as I mentioned, so anywhere from a handful up to maybe 100 at most are involved, okay? There are also shorter trials. The number of trials for Parkinson's that successfully completes a phase 1 is about 65%, okay? So a lot, but not all of them, right? So then you have phase 2. So they look at a much larger group of individuals. So we're talking hundreds up to maybe 1,000 or 2. And this will have the patients who have the condition, Parkinson's, right? And so you will have, often this will have a placebo controlled arm, or if not just placebo, then standard medication treatments to compare to. And this is when they start to look at effectiveness. So there's not enough participants in the phase 1 trial to really say definitively if something's effective or not. You need more people. And so phase 2 is looking at effectiveness, but also still safety, right? And you don't want to make a bet on a medication with only testing it in 100 people, right? That's not very many. So phase 2 trials are a little bit longer. So instead of, up to maybe a year or a little bit more, we're talking a year to maybe 3 years, depending on the purpose of the trial. All right. Then you can go on. The success of phase 2 trials is about 35% make it on to successfully meet their endpoints to keep going. So now we're down to about a third of all the trials that come out of preclinical testing. Okay. So then you have phase 3 trials. So these are huge trials depending on, again, what they're trying to do with the intervention. So for example, let's say a migraine trial. Well, migraines are very different from person to person. So you may need 5,000 people to determine if a migraine medication works. Well, for Parkinson's, phase 3 trials typically are, in the several hundreds to low thousands, okay? They last longer. So they want to make sure that the treatment effect continues, okay? And that there's no delayed side effects that may come up 3 months, 6 months after the trial ends. And so with this, again, about 33, so less loss of successful studies, make it past that. So once they have data that proves that the investigational treatment works, then they apply for FDA approval of the medication. So this can take a while as well. So it can take up to a year, sometimes longer. The FDA may go back to the company or research group that came up with the investigational product to say, hey, we need more information on these side effects, or we're not convinced enough of the benefit. We'd like to see X, Y, Z. And so then they have to go back and either reanalyze their data or do additional studies. Okay. So then you have phase 4. So phase 4, most people don't hear about because it's when the medication or treatment is already available and FDA approved. Drug companies and device companies keep logs of people who report in side effects to see. So you can imagine that this is the final litmus test for a treatment, is when it's open to the full population and very large numbers are undergoing that. Any questions about the research pipeline? All right, so we're going to shift gears. So this is a picture of James Parkinson. And so he published first on Parkinson's, which we know now, that's what we've named it after him. But he called it the shaking palsy. So he had an apartment that overlooked a very busy street in town, and he had a balcony. And so he would observe people on their way to and from work or whatever their daily activities were. And he would notice certain people very characteristically would have a more stooped posture, possibly some shaking in their hands or head, and very slow. And so he described this in 1817. It wasn't fully recognized as a disease entity until Charcot in 1861 created the constellation of symptoms and named it Parkinson's disease. So you can see that it took about 100 years almost for there to be an effective treatment once this condition was fully kind of described and being able to be diagnosed. That's a long time. And so in the mid to late 1960s, levodopa was created. And so individuals who received it woke up. They weren't as stiff and slow. Their voices were louder. their tremors improved. But this medication, without being paired with carbidopa, made people sick. And so, you know, it was one step in the right direction, but we've have a long ways to go. All right. So Parkinson's disease is pretty common. So it's the second most common neurodegenerative condition in the US, right after Alzheimer's. This is only going to continue to be true because of our population is aging. One of the most reliable risk factor for developing Parkinson's is age. And so I don't know about you, but I can wish all I want that I'm still celebrating my 35th birthday every year, but time marches on. So there's really not much you can do to avoid this. And so you can see that very rare, less than maybe less than 0.5% of individuals in the US may develop Parkinson's in their mid 40s, late or early 50s. But then when you get into nonagenarians in their 9th decade of life at age 85 or older, it's about 4% or 1 in 25. That's pretty common. Yes.
Onset, or is that when it's diagnosed? I know many of us have had symptoms for a while, but
Yes, great point. This is from the time of diagnosis. So I want to shift gears and then talk about what changes are happening in the brain. I'm going to try to keep this simple, but feel free to interrupt me at any time and ask questions. So Parkinson's affects a particular part of the brain. It's very deep in the brain, and it's called the basal ganglia. Okay. And the basal ganglia is involved in the control of motor movement. So how the speed of movement, the fluidity of movement, coordinating how fast, slow things are. And so a chemical is, sorry for those who are online, I'm sorry. So a key chemical that helps regulate this movement is called dopamine. And so dopa, yes, that is basically what your levodopa is a supplement, a dopamine. So the key chemical is produced by brain cells. And that's what I was referring to earlier, is that the dopamine producing brain cells, the terminals are about 60% lost by the time you develop motor symptoms of Parkinson's. All right. So dopamine is a chemical messenger, meaning it can be sent as a signal from one brain cell to another. And then this is very simplified. There's a circuitry that has thousands of brain cells continuously communicating back and forth to control your movement. But you can see that this would be very key if you're not able to communicate with each other. Try organizing movements. All right. So it's a little repetitive. So what causes Parkinson's disease? The short answer is that we don't know yet. Okay. Likely, it's an interplay of different risk factors. So you have some genetics. So that's DNA that you inherit from your parents. Environmental factors, so exposures to toxins or infections. And then we talked about age. So with genetics, let me see, there we go. There's many different ones that have been discovered. So this graph shows on the horizontal axis, let me see if I have here, how common the genetic mutations are. So if they're here closer to the vertical axis, they're very rare, okay? If they're more out here towards the common, then their incidence is much higher. Then on the vertical axis, you can see the risk, which they're trying to overly simplify how this genetic mutation interplays to cause Parkinson's. So you can see up here, it says very high risk and causal. That means that if you have mutations in these genes, that most likely you will develop Parkinson's, okay? Down here, you have low risk. And you can see that these are more common. And so some of these mutations here you can have, and it puts you at risk for Parkinson's, but it's not sufficient to cause it on its own, like some of these. There's different ways that genetics can be passed along in families. And so there's different modes of inheritance. So dominant or blue, so you have the LARC2 and the SNCA up here, are passed along generation to generation. There's no skipping it. If you have a 50-50 shot of whether you inherit that mutation. Then you have recessive. So there's about 1 1/4 chance that you will inherit that. Okay. Some of these you need 2 copies. So then it's 1 out of 8 chance that you would inherit a mutant copy of these. All right. And then again, these are, they say in green, I don't know. That's a generous green. That these are just at risk. So they're not in themselves necessarily causal. So just to summarize, multiple factors lead to Parkinson's. So we talked about the genetics. Then we talked about some environment. So toxins like pesticides, having head injury, or chronic traumatic encephalopathy, or CTE, is a popular hot topic in sports. So with football players having repeated concussions, then they may be more at risk for neurodegenerative conditions. So that's environment. Then you have what's called epigenetics. And so it's not the actual DNA, it's not the instructions that we inherit that tell your cells of your body what to do, but rather things that can modulate it or change it to say, hey, make more of this protein, or whoa, calm down, let's stop making this protein. All right. So it's a balance between the two. So risk factors and protective factors. You'll see here, I reassure myself every morning with my caffeine intake that I'm helping to rebalance my risk. We do not recommend starting smoking. So you'll see that on there, but that is not a recommendation, nor if you have Parkinson's, clearly you're not going to reduce your risk if you already have the condition. And yes, I have been asked that question more than once. All right. So alpha-synuclein is the abnormal protein that those dopamine-producing brain cells have that are toxic, okay? Alpha-synuclein, when clumped together, makes the cell dysfunctional, and then eventually that cell will die. They will clump together, the proteins will clump together, and then they'll create these little balls of what they call Lewy bodies, okay? And again, this is what it looks like on pathology slides. So it's probably been a long time since anyone got a microscope out and looked at pathology slides, maybe high school, huh? Okay. So this is, let me know if I'm getting too much in the weeds here, but I want to go through how alpha-synuclein is made so that you can understand where the clinical trial targets are, okay? Oop. You guys got a sneak peek. All right. So this is where your DNA is stored in a brain cell. So this is a very simplified picture or diagram of a brain cell. And this is the command center. This is where your DNA is stored and where the directions for how brain cell parts and functions are those orders are given out. So you have the alpha-synuclein gene, and it is then copied into instructions. So you can think of it as someone giving orders or writing down a recipe or instructions and handing that off. So that set of instructions then leaves the command center and goes to a different part of the brain cell that has machinery to assemble protein. So it has the instructions on one side, and then it has all the parts to make different types of protein. And so based on the instructions, it will assemble a protein, alpha-synuclein. All right. So this is a normal, or a depiction of a normal alpha-synuclein. And then you have abnormal alpha-synuclein here, kind of squiggles all clumped together. So the instructions, depending on their integrity, meaning if the recipe is correct, you will get normal alpha-synuclein. If the recipe was flawed based on DNA or the blueprint instructions, then you may get the abnormal alpha-synuclein. Sometimes, even with a normal recipe, you can make a mistake, right? So rare, but it can happen that you have some abnormal alpha-synuclein proteins that are made, but your body is equipped to clear. Okay? So a healthy brain cell can take the abnormal protein and clear it. So you can think of this as the cleanup machinery or the garbage disposal, however you want to think of it in your mind. And so it says your body identifies abnormal, says, got to get rid of this. And so there's different ways it can be broken down. All right, alpha-synuclein's exact function with the normal protein structure isn't fully fleshed out, but it's thought to help the transmission of those signals from one brain cell to the other. So, having zero alpha-synuclein is not necessarily a good thing, right? So you want to have some in the normal protein structure. All right, how many people have I lost so far? Okay. All right, so what happens in Parkinson's disease? So eventually you'll make the abnormal alpha-synuclein here. So this problem can arise from many different situations. So it can be that the DNA blueprint was wrong. So if you have the wrong instructions to begin with, that's going to cause a problem with your protein structure. The problem can occur on when the protein leaves the command center. You can imagine, the phrase, don't shoot the messenger. Well, if the messenger doesn't get there, then that's a problem too, even if your recipe or instructions was perfectly fine. Then you have the machinery that assembles the protein. So it takes the instructions and it takes different building blocks to create the protein. Well, if that part of the machinery is dysfunctional, you're not going to build the right structure either. All right. So, and then here, your fail-safe, your cleanup or garbage disposal may not be able to identify the abnormal protein and clear it from your brain cells. And so it can remain in there and cause toxic effects. Alpha-synuclein, the misfolded ones, can clump together and form these large fibrils that then are stored into the Lewy bodies, those kind of pink ovoid shapes I showed you on the pathology slide. Alpha-synuclein can be sent out as a messenger between the cells. And so if that happens, it can be taken up by neighboring brain cells, and it can actually trigger the normal alpha-synuclein in the other brain cell to start folding the wrong way. and start clumping together. And so you can have propagation of this abnormal protein clumping. All right. Let's take a breath. All right. So again, I moved here from Dallas. I think I left when it was just a point under 100, not with the heat index and humidity. And my kids were ready to spend the summer in indoor camps due to the heat-related illness risk in daycares. So when they arrived, this is the day of arrival in our neighborhood park. They were very happy. They jumped out of the car and they were so excited that they could see some mountains instead of a concrete jungle of Dallas. And so they've had fun ever since. So that's a little bit about me. That's my son, Oliver, who is now 4 years old. He would want you to know that. And my daughter, Evelyn, who's 7. All right. Right back into the science. Okay. It was a small break. It wasn't an exercise break, but here we are. So there's different ways that we can target this process in Parkinson's disease. So we went into great detail so you could see all the different ways that we can change the outcome of Parkinson's disease. So this is really hopeful. And it's also really helpful to know when you're going to look at clinical trials, kind of how they work or what's the idea behind it. Okay. So starting back at the command center. So you have DNA. We talked about how the DNA, the blueprint, could be wrong, right? And there's a trial that's ongoing for a particular genetic mutation that will correct the DNA. And so it'll enter into the command center and correct it so that it can create that recipe or set of instructions for the correct protein structure. That's really cool. All right. Then you have transportation outside of the cell, or outside the command center, sorry. So again, if the messenger never shows up with the instructions, that's a problem. But there's ways to help that messenger successfully complete their mission. Also wanting to make sure that the machinery or the assembly factory is up and running to create and complete that appropriate protein structure. There's ways to, oh, excuse me. There's hope. That was the last slide. Okay. No. All right. So there's ways to actually discourage proteins once they're made from taking on an abnormal shape and clumping. And so we can stabilize the normal protein structure. Okay. We can also take the abnormal proteins and then encourage them to say, hey, you got this structure wrong. You're supposed to look like this. And so that can be helpful as well. If you do have abnormal proteins, clumped alpha-synuclein, then you can help your brain cells identify them. So there are compounds that will help identify and label this abnormal protein to go in the garbage disposal. Now, some parts of the brain cell that can be dysfunctional and lead to Parkinson's can be the cleanup crew or the garbage disposal itself. And so there are plenty of targets and some ongoing trials right now to correct this. So even if your body is completely normal up until here, and then some abnormal alpha-synuclein accumulates, your body doesn't have the natural pathway to clear it. This is also a novel area that no matter what's causing your Parkinson's, if you can clear the abnormal protein before it can transfer to another cell, then that would be good too. So it's good specifically for the genetics that leads to the garbage disposal problem, but it's also good if it's being caused by another issue. Then you have over here where the abnormal alpha-synuclein is being put into these little bubbles to be sent to the next brain cell as a communication. Well, if you prevent this from even leaving, that's another great target, right? So only this brain cell would be affected. It can't spread it to its neighboring friends. Then let's say it did get spread, and this is the kind of very tiny, teeny tiny space between brain cells, that if you have an antibody to identify it and clear it, then it can't be taken up by the next neighboring brain cell. So there are many ways to target reduction of alpha-synuclein protein. And so again, there are many ongoing trials for various points in this process. There's also, remember, many different types of trials. So I spent a lot of time here at the end talking about this pathway because this is what we consider to be more disease modifying, to slow down progression or maybe even stop it, okay? There are symptomatic treatments. A lot of the symptomatic treatments will encourage dopamine release of the brain cells that are remaining in Parkinson's, or help that communication of dopamine as a chemical signal to work better, or to enhance that signal on the neighboring cells. So it's not just disease modification and alpha-synuclein targeting to make sure that you only have the normal protein structure. There are other non-disease modifying symptomatic trials as well. So I don't want you to forget that. All right, that's it. That's a great question. So for, let's say, the genetic conditions that may put you at risk for Parkinson's, the command center blueprint is incorrect, or your cleanup crew garbage disposal isn't working. And so those are different genetic mutations. So we can test for those genetic mutations to see if those targeted therapies would help an individual. Now, there's other kind of ways to look at it is that there's more broad disease modifying properties. So even if your cleanup crew or garbage disposal system is working, could we make it work better or faster? And so those are targeted therapies, but that many people potentially could qualify for. And I don't think I repeated the question. The question was, how do we know if someone is a candidate to receive an investigational drug that would target any one point in that alpha-synuclein process? Okay. Other questions? We'll go.
I noticed the slides back. It said high risk dairy.
Mm-hmm.
I'm a big dairy. What does that mean?
Yeah. So the question was, on one of the earlier slides, and whoops, one of the components on the balancing act of risk, at risk, or protective factors was dairy. So there was a study done in the Hawaiian Islands of individuals who were able to drink dairy on a frequent basis versus those who did not. And they found that the individuals who drank dairy had a higher, incidence of developing Parkinson's. Now, this has been kind of a one-place study, and there's different compounds in the volcanic rock that is also influencing the grass that the livestock eats. And so it turns out there's probably much more to it. And so I absolutely do not discourage anybody from drinking dairy whatsoever. Maybe not from Hawaii. Great question. Yes, sir.
Way back, and you don't have to go back into the slide, but in the research gathering, we started out with an age group relatively young and going up to the 80s. And how do you gather the information on somebody, or how do you determine somebody has Parkinson's or even the symptoms of Parkinson's? And it could take 10 to 15 years onset, which the person is not aware of until you finally go to a physician 10 or 15 years later and say, you know, what's going on? And he says, you may have Parkinson's disease. And by now I'm 80 years old. So wouldn't there be some kind of a skewed information, if you will, for the lower age groups?
Mm-hmm. So the question, yes. So the question was on the epidemiology slide. Let's go back to it. If there's basically a lead time bias, so that maybe more individuals in their 40s and 50s have Parkinson's but aren't having symptoms of it yet, I think is what you're getting at. Whereas now, by being in your 80s, you've declared yourself. So that may be, you know, epidemiology studies are limited by where the science is at. So our diagnostic criteria, either clinically or more recently, based off of, you know, imaging tests, biopsies, other more research-based ways to track people that may be at risk for Parkinson's. And then they start having what we call biomarker evidence of Parkinson's disease, of that dopamine-producing brain cell loss, even before they have symptoms. But, you know, really, for this information, it's based on clinical symptoms. But you're right, it could be skewed. But I think the proportion of individuals living with a diagnosis of Parkinson's, again, is much more prevalent in the older age.
Just a follow-up to that. Most of us here are probably recently diagnosed. I had a very serious automobile accident where I had traumatic brain injury, which I found really exacerbated the symptoms, which caused me to go to the doctor to say, what's going on? And he finally took him a long time to finally say, other than you have tremors, to say, yeah, probably have Parkinson's.
Okay. So the question, just to repeat it for our online audience, was, you know, if somebody has a serious head trauma event, it seemingly made the symptoms of Parkinson's much more prevalent or obvious, and hastened the diagnosis with a physician that was already following the patient for Parkinson-like symptoms. Is that?
Well, he was following after I had that accident, I really started to notice the tremors,
the shaking and stuff. Yeah. So head trauma, it depends on the extent and how many times your brain has had trauma to increase your risk or to contribute to the development of Parkinson's. So anytime that there's head trauma, there's a chance of brain cell damage. And as we've gone through extensively, if your brain cells aren't functioning properly, there's many points at which you may be having that abnormal protein structure, then leading to Parkinson's disease. All right. There's also very commonly we hear the story of, oh, I had a hip surgery, and then, you know, as I was recovering, I noticed that all my Parkinson's symptoms started. Well, that's likely untrue. They probably were much more gradual, but you were able to compensate for them. And then you had a setback that you didn't have as much reserve to compensate for, let's say, your slower movements or your stiffer muscles. And so things you were not able to do as well after the surgery or the trauma as you did before. And so a lot of people will kind of have that snap to grid phenomena of, oh, maybe there's something happened in the surgery that caused my Parkinson's, which is usually not true. Okay. Other questions? All right. Yes, that's a great question. So the question was, is there a website that goes over the different clinical trials, the results, or the progress of them? So there's many different websites. The Parkinson Foundation is one. Then you have Michael J. Fox, so they will do news blasts with good research updates. Different companies who are developing medications and drugs for Parkinson's will also have information on their research pipeline. And of course, at the very end, I was told I could put a very small plug at the very end. Oops. All right. So we are one of many places that you can participate in clinical research or find out more information. And so this is a QR code. So if you open your phone as if you're going to take a picture of it, it will have a yellow button with a link that you can click on. It will take you to the research website. There you can find navigation bars for different ongoing trials. You can also much more easily, for those who are live in the room, stop by and talk to some of our lovely research staff. And so I'm going to give a shout out to Mon and Susan in the back. Not only are they great at boxing, but they're also very knowledgeable about the different studies that we currently are enrolling. If you want to stop by. Again, seeking out information, you are not obligated at any research site to participate. You can go as far as even signing the consent form to get all the information. And then you can decide at any point, even if you started the medication, this isn't for me. We don't want you to do that, but you are in control, okay? And I want you to feel empowered that you can seek out information, educate yourself about opportunities without being obligated to participate. Yes, sir.
Is there an age out where they can you age out of it?
So that's a great question. So do you age out of clinical trials? For some, you do. So often 80 or 85 may be the upper age bracket accepting in trials. There's other trials like natural history trials, biomarker trials, symptomatic treatment trials that may be able to enroll older. But usually 80, 85 is the more common cutoff. Good question. All right, Jetty. Great question. And I'll repeat it. So, if you come see me in clinic, I will recommend, very similar to the presentation before me, to do about 30 minutes, 5 times a week, combination cardio and light weightlifting. It actually hasn't been definitively proven that higher intensity, so more higher heart rate, more weight, has been beneficial as a whole for Parkinson's. Those studies actually showed a higher fall risk or fall rate for some individuals. Now, that all depends on who the study was enrolling, right? So if you're enrolling people that are already falling, then yeah, they're going to fall more probably if you have them exercise at a high intensity. Whereas if somebody has very good balance, then they may be able to tolerate some of those higher intensity. In terms of exercising more in time, or spreading out into doing other things, it really isn't well established that you would have additional benefits. Being committed for 30 minutes 5 times a week is pretty darn good. So I would start there. Okay. And although, one more thing on the exercise. So, we have really good animal model data in Parkinson's, and we have good human data in Alzheimer's disease, which is another protein clumping disorder. The definitive human trials for Parkinson's and exercise to change the disease course, it takes many, many, many years to determine if it's actually, doing that. And so a lot of those are still ongoing, but there's many shorter studies that are highly suggestive that exercise can slow down disease progression. And I know myself and movement disorder colleagues pretty much 100% in agreement that this is the one thing that we have that can slow down disease progression. Great. Other questions? Concerns? All right. I love talking about Parkinson's, and I've got 4 more minutes, so don't leave me hanging if you got a question. Yeah. So exercise helps the integrity of brain cells. And so with routine exercise, the ability to clear toxic chemicals, not just alpha-synuclein, but other like reactive oxygen species and things, can improve significantly. So your cells are able to take out the trash more efficiently. They're also able to undergo stress a lot better. So let's say you have a brain cell and its neighbor tries to send over an alpha-synuclein abnormal protein, then it may have more defense mechanisms, those sorts of things. Yep, great. Thank you so much. I appreciate you inviting me to come speak.